Hi, this is Miss Linton. This is our third big review, and this is our second big idea. So big idea one, what was, if you were going to give me one main thing about big idea one, what would that be?
Evolution.
Evolution. And if you're going to give me one thing about big idea two, what would be the word you would pick out of there? Homeostasis is a pretty good word. What else could you pick? Energy. Good. Now, if this is the first time that, oh, did I say hi?
Hi.
If this is the first time you have joined in the big idea reviews, starting at the big idea one, remember I have a document for you. It has all the big ideas in it, associated with all the enduring understandings, yes, and the 55 things you need to know. And then I have another document with the learning objectives. So we're working our way through that. So big idea two, how we utilize energy and molecular building blocks to grow, to reproduce, and to maintain homeostasis. You know what that reminds me of? Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. That was weird. I hope he's okay. All right. Okay. So, first enduring understanding under this one, 2A, growth, reproduction, and maintaining organization of living systems, and here's the big part, requires energy and matter. I wonder what we'll be talking about. I know, energy is the capacity to do work. We know that. Laws of thermodynamics. You know the first law. Energy is neither created nor destroyed. It can just change forms, right? Ultimately, where does all of our energy come from? The sun. Good. All right. And then number two is that every time energy changes forms, you lose a little bit as what? Heat. Heat is not useful energy. Heat is not useful energy. Okay? Now, entropy discusses disorganization. All of us go to greater and greater disorganization, and the whole reason we do cellular respiration. Who does cellular respiration?
Oh.
Exactly. The whole reason we do that is to fight this. Right? Now, to the sun's credit, it's undergoing entropy too, right? And in its process of entropy, it's releasing energy. Do you agree with that?
Yes.
Okay. That's great for us because that energy travels in waves to us here on the earth, right? And we can use that energy that it gives off in order to change that energy into a form that's useful to ourselves, and then we can use that to fight entropy in our bodies. So then we talked a little bit about a low entropy system. So you have to enter energy into our bodies in order to have to avoid entropy. Now, who has more potential energy, messy kitchen or clean kitchen?
Clean kitchen.
No, you have, this one is the harder one to maintain, right? And this can't get any worse than this, right? Okay, when you have something that's complex and organized, I know, like putting hydrogen ions on one side of a membrane. Have we looked at examples of that? When you have low entropy, high organization, you can use that to serve some purpose. For instance, when we concentrate hydrogen ions to one side of our mitochondria, we can allow those hydrogen ions to come from the intermembrane space back into the, do you remember the structure of the mitochondria? Matrix. So we can make some ATP. A lot of it, a lot of it, a lot of it, right? And chloroplasts do the opposite, right? They concentrate hydrogen ions to the center of their thylakoid membrane. So when it goes back out, right, you can use that to do what? Reduce NADP, right? You take some ATP and some reduced NADP to the dark reaction. Let's think about neurons. We concentrate sodium to the outside and potassium to the inside. Because when we have a reversal of charge by allowing sodium to come in, we can get a message from the dendrites down to the axonal bulbs, right? So that is a process of making something organized, putting it all together, it has greater potential energy than if it's dispersed. If I have heat in a small, closed, confined position versus, no. If I have heat in a small, confined, closed position versus dissipated, it's more effective to have heat in a small, closed, confined position, right, than dissipated. Just think about if you used a magnifying glass and you're being mean and killing ants, right? You can concentrate that energy in one spot. It's more useful. Do you have the big picture of that? Yeah? Okay. So when we look at this diagram here, this is more organized. This is glucose. This has more potential energy because of all the bonds between the carbon and the hydrogen than this less organized carbon dioxide and water. So this has more potential energy. Which has more entropy? The glucose or the carbon dioxide and water? Carbon dioxide and water. It's more disorganized, right? Okay. Here, like I talked about before. Now, somebody next to you, why don't we have the person in the blue seat? Why don't you explain this, since I've already gone over it once. So you put it together. And if you don't have a bio buddy, hook up with a bio buddy. Go.
Okay. So we'll look at the money. Okay, so we see that at the first side is not so but since the one side
all right, so if you have more hydrogen ions on one side than another, we can use this. Maybe we can allow these hydrogen ions to go through some ATP synthase complex, perhaps, in order to synthesize ATP. We remember that. All right, so delta G, that's Gibbs free energy equations. Yay! Free energy is the amount of energy that is free to do work. So exergonic reactions. What do we hear in exergonic? Exit, right? If it's an exergonic reaction, it goes from high energy to low energy, right? So the delta G is a negative number. More, so less, okay? Delta G is negative. Those types of reactions are more spontaneous, right? Whereas if you have an endergonic reaction, you're going from low energy to your products having more, higher energy, right? So that's an endergonic reaction. We have to put energy into that. Irregardless, these types of reactions, you're going to probably use an enzyme to act as a catalyst for that reaction, yes? And that's especially important when you even in an exergonic reaction where you're losing energy, it's more likely to happen. Hydrogen peroxide, if given time, will break down into what? Do you remember what hydrogen peroxide will break down into? Water and oxygen. We could see the oxygen bubbles bubbling up. We could measure that oxygen that was generated. Do you remember that part? Okay, so that can happen spontaneously, but it would happen better if we had the enzyme catalase. It's still an exergonic reaction to do that. So there must be a hurdle that prevents that reaction from going forward. What's that hurdle called? Energy of activation. And enzymes will do what? Lower that hurdle. Okay, just kind of sticking that in your head. So when we look at Gibbs free energy and measuring that, we have enthalpy, heat content, that's the energy that you have in the bonds, right? We're taking, what energy do I have in the bonds? Lord. It's okay, baby. Child number 2. Man down, man down. Okay. I'm saying how much energy do I have in the bonds minus temperature, right? Times entropy. Can think of as randomness or breaking down. That's how we calculate it. How much energy do I have in the bonds minus the energy time, the temperature times the entropy. That is Gibbs free energy. I would like to continue, please. Continue. Okay, thank you. All right. So when we look spontaneous, if our delta G is negative, if our everything okay?
Yeah.
You gotta go too?
Yeah.
Okay, later, Gator. Weird things are happening. Some weird things. In case you don't know this, if you take a big number from a little number, right, then it's going to be what? Negative. What could make this number big? The change in the entropy, right? This number could be big because the temperature is high, and this is going to be a negative delta G. It's going to be more spontaneous, right? You're going to release more of that energy with a higher temperature. Does that make sense? Okay. So we talked about this, exergonic versus endergonic reactions. There's a little hurdle right there, and an enzyme will take that black line and turn it into the red line and lower it, make it more likely for that reaction to occur. It does not, an enzyme does not change your ending point where you're going to end up. It just makes it more likely that you will get there. Okay? The delta G is still the same. ATP. This is the energy currency of our body, right? Adenosine triphosphate. There is a big bond right here when this energy is released, 7.3 kilocalories per mole, I believe. I can't remember 100%. I think it is. Then that's the amount of energy that is released when you break that bond. So you can't break part of an ATP. You've got to break all of it. So if it ever asked you a question about that and you needed, let's say you needed 17 kilocalories, I don't want you to say, oh, I need 3.2 ATPs. You don't do that. You would have to, if you need 17, you're going to have to break down 3 ATP molecules. You follow me on that? You can never do part. It's either broken or not broken, like bells that we can't unring, yes? Okay, and so then it'll release that energy down to ADP. You can even break this one down as well. You can break it down and make it be AMP as well, but we don't typically do that in our cells. Creating a hydrogen ion gradient, then you can use it. So use energy, and we talked about in cellular respiration and photosynthesis, you needed energy to get those hydrogen ions to the outside. In photosynthesis, what did we use for that energy to get those hydrogen ions to come to the interior of the thylakoid membrane? What did we have to use? Energy from the sun, yeah. The sun, we use that energy to excite, right, the electrons from what? What electrons? From water. Those electrons got excited by the energy from the sun, and as they came back down in their energy level, they transferred some of that energy in order to pump the hydrogen ions to the interior of that membrane, right? And then when they wanted to go back out, they could make ATP. Where did we get the energy to get the hydrogens to the outside when we talk about cellular respiration? Where did it come from? Reduced NAD. And where did the reduced NAD get that energy from? What? Oh, okay. But where did those come from? Ultimately, where did it come from? Yeah, the food that you eat, right? Right? Remember we take glucose, what do? Break it in half. How many steps? 10 enzymes, 10, right? 2 pyruvic acid, right? All right, 2 plus 4 is 6 is a bit, yeah. What did you make? You made some incidental through substrate level phosphorylation. You made a little ATP, right? Substrate level phosphorylation got us how many ATP in glycolysis? 2. Substrate level phosphorylation got us how many ATP in transition? 0. How about in the Krebs cycle per glucose molecule? 2. 1 a turn, right? So we didn't get a lot of ATP from that, right? But we did form a lot of reduced NAD. And they would take them to the top of the ETC, which was the electron transport chain. They dropped off their high energy electrons, whereas the sun got the electrons high for plant, right? And and then we use that hydrogen ion gradient to concentrate our hydrogen. Remember, reductase, cytochrome oxidase, to concentrate those hydrogen ions on the outside, which allowed them to come back in through ATP synthase complex, right? You're remembering all of that? I'm just trying to get you to see the big picture between energy, right? Energy, how we acquire it, how we get it, and then how we use it to build things within ourselves. So we needed to understand the idea of redox reactions, which is when one compound is getting oxidized, you're going to have another compound that gets reduced, right? So, for instance, in cellular respiration, when we're dealing with glucose, what are we going to do ultimately to glucose molecules through the process of cellular respiration? We're going to what them? Oxidize them, right? We're going to oxidize the glucose. How can you oxidize something? It could what? Gain oxygen or lose hydrogen or lose electrons, right? So if they're losing their electrons / hydrogens, and those electrons, hydrogens are going to who? NAD, right? Reduce some NAD, right? So you're taking the electrons from the food, electrons / hydrogens from the food, and giving it to NAD, NAD is taking it to the top of your DTC. It lets them go then, lets them go ahead and drops their energy level. And in the process of dropping their energy level, you create a hydrogen ion gradient. Then you can harvest that hydrogen ion gradient to synthesize ATP because it's going to want to go from a higher to a lower. And we call that change across the membrane. Remember we talked about the chemiosmotic potential, right? Because we had a difference in concentration, charge, and pH. Very motivating to want to go from the higher, because everything goes to greater entropy, right? So that's low entropy when they're all on one side, and they want to go to greater entropy, and that's why they go to the other side. Yes? And we can harvest that entropy breakdown, right? That increase in entropy. We can use that to synthesize, that happens at the same time, right? To synthesize our ATP, which is then good for us. So you can see from the big picture, it's a series of one thing breaks down, and while it's breaking down, right? Oh, darn, it broke down. But somebody else can use the energy that's given off by that broke. Sun breaks down, we use that breaking down to build our own structures. You see the big picture is what I want you to see there. Okay, next, this is looking at a cross-section of plant and a leaf. You can see the chloroplast. What is the function of the membranes inside the chloroplast? Why are they even there?
Increase surface area.
One is the increased surface area for the reactions to take place. What else? We need to create structures so there's a difference across the membrane. So we have a chemiosmotic potential. So by having these thylakoid membranes, having these grana in here, all these stacks of like hollow pancakes, right? Then we can concentrate the hydrogen ions on one side of the membrane so it's different inside the grana than outside, right? We're concentrating those hydrogen ions in there, so they want to go from a higher concentration, low entropy, to higher entropy, like, right? They're going to move out, and then we can use that as it moves. It goes through ATP like a little turnstile in order to make ATP. So you need the compartmentalization that the membranes provide, so you can have a difference across the membrane, just like you need a difference across for a neuron or a difference across for the mitochondria. As long as you have membranes in there, you don't have to have a chloroplast, right? There's plenty of autotrophs that are prokaryotic cells. In fact, most are. Most of our autotrophs are. But they just have to have a membrane somewhere. It doesn't have to be a closed-off organelle, but a membrane that you can have a difference across that membrane so you can use that energy in order to make your ATP. Doesn't matter if you're multicellular or unicellular. You need some sort of membrane to create that difference. Okay? Now, not all light is useful for a plant, because not all light will excite those electrons, right? That's why what light, if I was going to grow a plant, you'd say, Miss Linton, don't grow it in this color light. Green, right? Because if my jacket is lavender or purple or whatever, that's telling you it's what kind of colors can my jacket absorb? All but purple, right? That's getting reflected back to you. What does the black dress say to you? Yeah, it's absorbing all. So if Michael and I went outside, who would get hotter faster? I would, because I'm absorbing all the energy and he's reflecting it off, right? So not all energy, did you see him? He just reflected it, I saw him. Not all energy would be super helpful for a plant. And what determines that? The pigments that are allowed to capture that energy, right? Pigments are capturing that energy and transferring it to the ETC. And so what it's saying is they don't have pigments to absorb in that wave. Now, what if you're a red leaf? Then don't use red light, right? You would want to use what? Green. Now, I'm going to tell you, when leaves change colors, though, right, if they're changing colors, it's usually because the season's changing. So it's too cold and that chlorophyll is already broken down and you're using a different pigment at that time. Okay? Let's just turn on there. Okay. When we look at the summary of it, we can see water comes in. Hey. And it's like the weirdest day. It's just the weirdest day. I just want to tell you it's been weird all day. The water breaks down, right? Break it in half. What do we get? Oxygen and electrons. Now, is this oxygen a waste product for this plant? What else? What do they need it for? Cellular respiration, right? So they generate that oxygen. That's great for them because once this environment started being an oxidizing oxygen revolution, right, about 2.3 billion years ago, we were able to do aerobic respiration, right? And so as a result of that, that allowed multicellularity, right? Ozone layer, right? That tells you that a plant must do more than what?
Photosynthesis.
More photosynthesis than cellular respiration. Because remember when we talked about gross primary productivity and net primary productivity? Just tying everything in because now you know everything, right? Because you're like, oh, you're just jumping around. Okay? Gross primary productivity is all the energy that the plant takes in and uses to build its sugars, yeah? Net primary productivity is whatever's left over after they've met their needs. And they say, now you can have this. Okay? So that's net primary productivity after they've done cellular respiration. Okay, so water split, oxygen is given off. Light is exciting some electrons through photosystem 2 and photosystem 1. And as a result of that, you make some ATP and some reduced NADP. These electrons right here originated with who? Water, H2O. These electrons right now are going to get hooked up with who? CO2. So when you eat sugars from a plant, right, because it's going to produce sugars, in order to build those sugars out of the CO2 in the air, to fix that CO2 with, what's the enzyme that does that?
Rubisco.
Yes, Rubisco, RUBP, right? In order to do that, okay, it needed electrons from water. So when you're eating something from a plant, you are eating part of the water that went into that plant. Right? Because you use that to reduce the CO2, right, of that you're making. And then as a result of that, the reduced NADP that came in, it gave up its electrons, it got oxidized, and it used it to reduce the CO2, right? And now you can reuse that again, and you can reuse the ADP because you needed, it was an energy investment. You have to put energy in order to do photosynthesis. 2 reactions. I'm going to let whoever talked last time, I've been talking for a while now, I want the other bio buddy, talk about what's going on from here. Now, it really works right here, photosystem 1, and then photosystem 1 gets replaced by photosystem 2, and then photosystem's 2 electrons get replaced by water. That's the actuality of how it works, but you can still explain it from left to right. Go ahead.
Okay, well, as you can see,
This is Okay, I want to try to get through this enduring understanding, so I'm going to pick it up a little bit. Can you handle it? I know you can. Yes, you can. You remember this?
Yeah.
Yeah, we already went over this. So this is just the close-up of that thylakoid membrane, yeah? And so that's your light reaction. Here's your dark reaction. 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take one away.
PGL, G, 3P.
And your left with 5C3. Use some ATP to build 3C5. And da-da-da-da-da, and you make glucose, right? I'm happy to do those songs again with you, but that's from the light reaction. Then you do the dark reaction. We saw an adaptation right here. This is an adaptation for areas where there was a lot of oxygen. What is the problem when you have a lot of oxygen? It likes to hook up with RUBP instead of CO2. Remember we talked about RUBP as a garden tool?
Yeah. Oh, yeah.
Yeah, because it gets tempted by the fruit of another. So some plants have this what's called Krantz anatomy, where they isolate and they do their dark reaction, their Calvin cycle, their light independent reaction, these are around these bundle sheath cells that are around the xylem and the phloem. And then they have something called PEP, which escorts CO2 in because PEP will not hook up with oxygen. So the bundle sheath cells are only exposed to CO2, and it costs you ATP, it actually costs you some NADP, but in the long run, you get more product out of it than you would without. Okay, I'm skipping that because I'm trying to get through this big idea. Here are the steps of cellular respiration. We can go over that song. We know all of it, right? We have glycolysis. If you have oxygen available, you're going to do transition prep. Then you're going to go into your Krebs cycle, 2 + 4 is 6, do the bed, right? And then we're going to take all of our gather up all our reduced NAD and FAD, take it to the top of the ETC, and make some ATP. Okay. We know glycolysis, we know transition prep. We can see this is a good example of where you're taking the hydrogens here. We can see the CO2, and you can see the H on here. We're using those to reduce our NAD because we've got 2 of them. And we have our acetyl-CoA we can feed into our Krebs cycle. Again, I'm happy to do the song. You remember this stuff pretty good, right? Pretty well? Okay, and then we talk about the electron transport chain.
Yeah.
Are you feeling comfortable with that? We hit this stuff pretty hard, so just want to make sure. I'm happy to go over it again with you. And I don't know why that's back up there again. Oh, because I wanted to talk about fermentation. Now, where do we get our energy from? This ties into the digestion part. So if we're going to put sugar through glycolysis, we have to be able to get it. So we talked about 2 kinds of digestion, extracellular and intracellular digestion. Extracellular digestion, I put a star by that because it is more what? Adaptive, right? In the sense that it's more efficient. Because you have a tube, you put food in it, and you throw the enzymes in there, digest, and then absorb the nutrients out. Whereas in within the cell, intracellular digestion, you're doing it individually per each food vacuole. We talked about homeostasis, and we talked about how we access water, how we lose water, remember, our cells are trying to maintain homeostasis, but there's fluid around each and every cell. So even though we're multicellular, we need to think of our individual cells and how they maintain homeostasis. So we need the fluid around them to be providing the oxygen and the nutrients and to take away the CO2 and waste. And who's going to bring that? Because do we have an open or closed circulatory system? Closed. So our blood will, and we know about the differences between the hydrostatic pressure and the osmotic pressure to push out the nutrients and the oxygen, pick up the CO2 and waste if it's our systemic circulation, yes? But we know that all of these play a role because our lungs have got to breathe in oxygen, right? We know that we need to get rid of waste using our kidney, nitrogenous waste. And we know about our liver has multiple functions in order to help maintain homeostasis. We talked about the 4 important organic molecules, carbohydrates and what role they play, right? We use carbohydrates for both short and long-term energy and also structure in living organisms. What would be a structural component of a carbohydrate? Cellulose, right? A cell wall would be structural. Okay. What would be an example of one for energy? Glucose, simple sugars, glycogen in our liver, starch in plants, right? Okay, lipids, what are the 2 categories of lipids?
Steroids and fats.
Steroids and fat, right? We know about phospholipids, we know steroids and cholesterol, and also in hormones, right, in our endocrine system. Proteins we know we use. What are some things we use proteins for? Tell me. Muscles, hair, enzymes, exactly, and nucleic acids, right? DNA and RNA, right? So we use our energy, right? We can use this as a source of energy, but then we also use our body's energy to make the carbohydrates we need, to make the fats we need, to make the proteins that we need. This was a summary chart I made. I made you a much better one.
Yeah,
this is the ugly one.
Yeah, we have the better notes.
Yeah, it's on the notes. This is super ugly. Sorry, I should have updated that. Then we went into some of the systems on how we would access the different gases. So I'm guessing what cycle is this? Water cycle. We know about evaporation, transpiration, precipitation. We know about groundwater, right? We've talked about that. Here's our phosphorus cycle. Where's the reservoir for phosphorus?
Rocks.
Rocks. How do we get it out?
Weathering.
Weathering or man-made screws any of these things up, right? We know it as a limiting factor for algal growth, and when you get too many phosphates in there, then you can get an algal bloom. That's bad. Causes death of a lake. Why does putting a bunch of plants in a lake cause death of a lake?
Because when they die,
Die.
The fish die.
detritive force.
Decomposers, break them down, cellular respiration, then no oxygen, then the fish die. You got it. Okay. Nitrogen cycle. We know we have nitrogen fixing bacteria. Why do we need nitrogen? For our nitrogenous bases and nucleic acids?
Of course.
Yes.
All right. For our amino acids, right? The amino group?
Oh,
Why was
no,
there
no, no,
so
no,
much
no.
N-word?
Because N was
Yeah. All right. So we do we don't. Bacteria do nitrogen fixation, forming ammonia, nitrification, nitrite, NO2 minus, nitrate, NO3 minus. Who can use those? Plants. Animals eat the plants. Plants and animals die. Decomposers, ammonium, nitrite, nitrate. What's another way to make it though? Atmospheric fixation and industrial. And how do we return it?
Denitrification.
And carbon cycle, which we've just been going over, right?
Yeah.
So what are the things that fix carbon? Carbon dioxide. No, what kind of
Cellular
things fix?
respiration.
Listen carefully. Fix carbon, CO2. Photosynthesis, only one thing. Okay? What releases carbon into the atmosphere? Cellular respiration. Combustion. Decomposition, yeah. Boom. You did it!
Yay!
So we'll start it Enduring Understanding 2B. I'm going to tell you, this big idea 2 is so big that I actually have to break it into 2 notebook presentations because otherwise it crashes.
Oh,
So this is a big one. This is a big one. Maybe we should work on it a little bit tomorrow too. What do you think? And well, it's your time to look over your exam.
Yeah.
So I think there's an AP English.
Oh, you have a meeting tomorrow?
I'll still
That's at lunch.
Okay, we'll see if there's any, but we'll start, we're on what slide are we on?
We could do it at 1.
43. Okay, have a great evening, have some toast.